Hi, everyone. My name is Daniil Gataulin. I'm one of the senior research analysts covering biotech here at Chardon, where our vision is to help identify companies with high potential for investment returns based on creating real value for society through disruptive innovation. It is my pleasure to introduce Shankar Musunuri, the CEO, and Arun Upadhyay, the CEO, Chief Scientific Officer, to our virtual ophthalmology summit, where our initial focus is on wet AMD or AMD and diabetes-related ocular diseases. Shankar, Arun, welcome.
Thank you. Thank you for having us.
Yeah, so to start things off, could you please provide a brief overview of Ocugen and talk about some of the recent milestones?
Ocugen is a fully integrated, patient-centric biotech company focused on vaccines for public health and cell and gene therapies targeting unmet medical needs through courageous innovation. Our goal is not only bring these cutting-edge technologies to market, also work even harder to provide global market access for patients who need them. Recently, this year, we released a few months ago our preliminary results on our first gene therapy program, OCU400, targeting retinitis pigmentosa. And we did demonstrate favorable safety and also visual improvements based on mobility course as well as BCVA in patients in first two cohorts, low and mid dose. And we're anticipating more data coming out in third quarter this year. So that's a key milestone for our gene therapy program.
Got it. So as you alluded to your gene therapy approach, you're pursuing the concept what we call or what you refer to as a modifier gene therapy. So you're leveraging that in the RP and retinitis pigmentosa LCA program, but you also have a dry AMD program that's also using the modifier gene therapy approach. So first of all, can you explain how this approach works and its potential advantages over what we call more traditional gene therapy approaches?
I will let Arun take it. Go ahead.
Thanks, Shankar. Yeah, so when we look at modified gene therapy technology approach, it is more like, rather than fixing a single genetic defect, this approach more like works, fixing the imbalances which you see in the organ or tissue into the disease condition. So modified gene therapy approach is based on a transcription factor, what we call nuclear hormone receptor genes. And these genes are basically master regulator in retina, and they control basic physiological process within the cells, such as cell development, differentiation, metabolism, and other critical cellular function, which is essential for health and function of the retina. And the uniqueness of this approach, as I mentioned, it is not like we are not trying to fix a genetic defect, but more like we are trying to fix the cascading effect of genetic mutation or environmental assault, like in case of age-related macular degeneration, incurs on the retina, and how to kind of basically ameliorate or improve those bad effect of the salt by fixing the various biological processes. So that's how it is unique and differentiated compared to other traditional approach. Other advantage of this approach is that with one product you can basically manage large population of the people, whether either they are affected with the genetic condition, particularly in case of inherited retinal disorders like retinitis pigmentosa and liver congenital amaurosis or Stargardt disease, or other complex form of diseases like age-related macular degeneration, where you have multifactorial disease, where you have disturbances in multiple pathways like inflammation, oxidative, complement. So it can also work in non-genetic conditions which are complex and multifactorial in nature.
Got it. Thank you for that overview. And with the focus of this session on AMD and diabetes-related diseases, I wanted to first spend some more time on OCU410, which you are developing for dry AMD. So can you talk a little bit in more detail about the mechanism of OCU410, and specifically what is RORA and what is its role in dry AMD?
Good question. So RORA is our second candidate in modifier gene therapy platform technology. It's a second molecule. And it is also a nuclear hormone receptor, and it play important role in modulating the retinal disease. So what we know that this RORA molecule play a role in development and function of rod and cone photoreceptor cells within the retina. And also it regulates the genes and pathways involved in onset and progression of age-related macular degeneration. Specifically, if I look at the molecular pathway, it is known and established that RORA regulate the path, genes which are involved in photoreceptor development, such as opsin 1, SW, MW, AR3, and so. It also plays important role in lipid metabolism, which are linked to or known to be disturbed in the age-related macular degeneration conditions like apolipoproteins, ApoA1 and ApoC3 and so. It also regulates the inflammatory pathway, which are known to be, maybe initial driving factor for the development of age-related macular degeneration. And in addition, RORA regulate the pathways which are linked to the oxidative stress, like hypoxia-like condition, like HI1 alpha and oxidative response related machinery within the retina. And most importantly, what I think everyone in this industry is targeting, complement pathway. RORA also regulate the anti-complement protein, CD59, which plays very important role in basically breaking the complement complex, which lead to the degeneration or damage to the cells during disease pathogenesis. So this is how the RORA is differentiated from other molecule or other a target which has been assessed or evaluated by other company in dry AMD space. So in nutshell, if I have to summarize, sorry. So in nutshell, if I have to summarize, OCU410 is a molecule which targets the multiple pathophysiological pathway linked with the dry AMD, such as lipid metabolism, which lead to the drusen formation, which is hallmark of dry AMD. It also regulates improve the anti-inflammatory environment within the retina. It is anti-oxidative in nature and anti-complement in nature. So altogether, it targets multiple pathway related to AMD pathogenesis and potentially provide better, or you can say improved, outcome in these patients.
Got it, understand. So we do have a recently approved medication for geographic atrophy. Where do you see OCU410 being positioned, in the treatment landscape? Can you comment on perhaps target patient population? Are there any overlapping functions and any other potential benefits of OCU410?
Good question. Yes, like the one of the key benefit, if I have to look, and of course we have not assessed this molecule in clinic, but one key advantage could be that this is a one-time curative therapy, so you don't have to get multiple injection, which what we see for currently approved product. And so that's one hallmark. Related to the disease population, the currently approved product is primarily targeting the disease in advanced stage. But if you look at the mechanism of action of the RORA, okay, there is high likelihood that this particular molecule may provide added benefit if the disease can be intervened in the early stage, because that early stage of the disease is where all these cascading pathway, lead to the kind of degeneration and drusen formation. So this product has not only potential in providing some benefit in the advanced stage of disease like geographic atrophy, but it may have, play some role in basically preventing the progression of disease to the advanced stage. And and also like some of the things related to the safety, we know that currently approved product have some safety concern, like development of CNB, and of course like other safety concern like vasculitis, which recently we came across. So maybe our product being protective in nature may not have those safety concerns, but still like that need to be evaluated in the clinical development.
Got it. And since there is no clinical data for it yet, wanted to ask if you could share some highlights from preclinical studies that give you confidence about this therapeutic in humans.
So we as you know, like there are not a lot of well-established animal model, especially for the macular degeneration or dry age-related macular degeneration. And in industry, like we have been using variably different model depending on mechanism of action and potential, what you call clinical symptoms, like blue light damage models, some genetic models. So for this RORA molecule, we have done like a lot of in vitro and in vivo animal study. So for in vivo study, we used ABCA4 knockout model. And this ABCA knockout model has been used in the industry for macular degeneration, including dry AMD, as well as other genetic macular degeneration disease, that is Stargardt. So in this particular animal model, OCU410 demonstrated a good, I would say, efficacy. And that efficacy was on multiple structural as well as the functional level. So OCU410 was able to reduce the drusen formation or drusen-like deposit formation in ABCA4 knockout mice model. So it indicates that this molecule has potential to slow the progression of disease based on the drusen phenotype. In addition to that, we also observed that RORA delivery to the subretinal space was able to improve the retina function, which was monitored by electroretinogram, which is one measure to look at the retina function. In addition to that, we also saw improvement on some of the structural perspective of the retina from histological perspective. And this molecule was well tolerable in this preclinical mice model. In vitro study, we also assessed the role of molecule which I was highlighting before, antioxidative property in ARP19 cell, which is relevant retinal pigment epithelium. We also demonstrated anti-inflammatory role in ARP19 cells as well as the microglial cells, which are resident, you can say, cells in the retina. And this molecule was able to induce the anti-inflammatory or enhance the anti-inflammatory function of those cells. And also in ABCA4 knockout mice model, we saw the enhanced expression of CD59 protein, which is anti-complement protein and play important role in disassembly of complement complex and its effect on the retinal cells. And why CD59 becomes important? Because CD59 is also being explored by another company in the retina space, where they have been kind of in early stage clinically, it has been a validated target where they are seeing some benefit. So in nutshell, RORA basically in all the preclinical study demonstrated that it can work on the various multiple pathway linked with the pathogenesis of dry AMD.
Got it. And can you remind us how is it administered?
Subretinal.
Got it. Okay. And in terms of the status for the program, for the OCU410 program, I think you initially were guiding for the second quarter IND submission. Just wanted to see if you have any update on that.
Yeah, so for this program, we are targeting to get into clinic this year. So yeah, we are pursuing that, yes.
Okay, but can you say if the IND has been submitted already, or you're still working on that?
Shankar, you want to take that question?
Yeah, sure. IND, yeah, we've been, we submitted it. We're working with FDA. And as soon as we get clearance from them, we'll start dosing patients.
Understand. Excellent. And I don't know how much you share, but curious to hear some highlights potentially about the study design, including, patient baseline characteristics and some of the outcomes that you'll be looking at.
Yeah, I'm happy to take this question. So in this particular phase 1/2 study for we are targeting advanced form of dry AMD to begin with. So primarily patient with the geographic atrophy. And this study will have a two part, you can say. In the phase 1, we are going to do the dose escalation where we'll be having like 3 dose levels, low, medium, and high. And we'll follow the same strategy what we did for OCU400, n equal to 3 subjects in each cohort. And then based on that, we are going to get into expansion phase, but our expansion phase is going to be slightly different. The approach is that in this expansion phase, we are going to have a proper control arm, untreated, as well as a 2 arm with 2 dose level, which will be selected based on the phase 1 portion of the study. So primarily phase 2 expansion phase for OCU410 GA trial will consist of 1:1:1 randomization. It will be a masked study where we'll be assessing the 2 dose level of OCU410 selected based on the phase 1 portion of the study and with the untreated control.
Got it. Understand. And as you alluded to earlier about the vasculitis, that's been observed with the approved products Syfovre recently. Does that have any read-through to your program, to OCU410?
Of course, we have not assessed our molecule in clinic, so there's nothing much I think we can state at this moment. But based on like our understanding on preclinical study and non-clinical safety and toxicology study, we don't think that there's something which we need to be alerted at this stage.
Got it. Understand. Okay. I wanted to move on to and talk about OCU200, which is a transferrin tumstatin fusion protein. So can you, again, it's a differentiated mechanism, right, from what a lot of effort's been focused on recently among other companies. So wanted to understand more about the mechanism and potential advantages of that mechanism versus approved anti-VEGFs and potentially some of the gene therapies that are in development.
So OCU200 is a fusion protein, and it consists of two distinct domains, with their own unique set of functions. So it has got transferrin. Transferrin is basically a protein which we are using as a carrier, which facilitates the movement of molecule from intravitreal side of injection to the target side in the choroid and retina, okay, where molecule is supposed to work. And the second component of OCU200 is tumstatin. Tumstatin is a molecule which is derived from the collagen and has been shown to have anti-angiogenic activity. So tumstatin binds to the integrin receptor, and through that integrin pathway, it modulates various function within the cells, which are associated with cell proliferation, cell division, and the development of angiogenic-like conditions. So this is what all we have about if you look at the unique property of the molecule. So currently approved therapy, if you look at, primarily they target the VEGF pathway. They are anti-VEGF in nature. And this current therapy, they have certain limitations. We know that there are a lot of individuals either who are partial responder or to some extent non-responder to current therapy. So OCU200, based on the differentiated mechanism of action targeting a completely unique set of pathway, which are linked to the modulation of angiogenic pathway, cell proliferation pathway, especially in the actively dividing cells, becomes promising target. So we believe that OCU200 may provide benefit to those who are non-responder or partial responder to currently approved anti-VEGF therapy. Coming back to the gene therapy approach path, Of course, like another advantage for OCU200 before I go to gene therapy, it is designed in such a way that we believe that we may have to give the dose less frequently compared to currently approved VEGF therapy. And the reason for that is its conjugation with the transferrin protein, which may provide the better residence time of molecule at the site of action. In contrast to the gene therapy approach, of course, this is not going to be one time. So we will have to give them multiple injections. But the benefit is that current gene therapy approach are still targeting the VEGF pathway. And when you deliver anti-VEGF constantly for longer duration, we don't know how it is going to fare well, okay, when you have anti-VEGF, because our cells, they do require the VEGF molecule for their normal physiological function. So it will be worth watching like how the safety profile comes out for the anti-VEGF gene therapy approaches. But this is how the OCU200 will be differentiated compared to that.
Got it. Thank you. And so for OCU200, I think you're looking at DME as your initial indication. You submitted an IND for that earlier this year, but subsequently it was placed on hold due to some CMC questions. Wanted to ask if you can comment on what those questions were and what's your progress in addressing and resolving them.
So primarily the question on CMC related to basically agency wanted us to have a method ready, focused on the potency of molecule at this stage of the program. Normally in the drug development, you expect this kind of assay as your program progresses from phase 1 to phase 2, phase 3. Normally you try to develop full cell-based potency assay. So one of the agency request primary for this clinical hold was to implement a potency assay for the release of drug product before initiating the trial.
Okay, and do you expect the trial to start enrolling patients at some point this year?
That's correct. We are still on track and we'll be responding to the agency request sooner. And yeah, we are hopeful to initiate the trial this year. Yes.
Got it. And speaking of the trial, can you walk us through the trial design and similar what do you expect to show?
So this particular OCU200 trial is only phase 1 study, and the whole objective of a study is to determine the safety and tolerability of the molecule. So we have total of 4 cohort in this phase 1 portion of the study. In initial 3 cohort, we are going to have the dose escalation of OCU200 molecule, low, medium, and high dose. And based on this dose escalation portion, we will determine the maximum tolerable dose of OCU200, and then we'll select that dose, and then we'll combine with the currently approved anti-VEGF therapy, and we'll do the additional 1 cohort trial. So we have a 3 cohort, low, medium, and high for OCU200. And based on those outcome of those cohort, whatever is the maximum tolerable dose, that dose will be chosen and combined with the anti-VEGF in the cohort 4, and it will be assessed. So primary objective is safety and tolerability, but of course we will be collecting some efficacy signal as well. And it is not single dose, so we are assessing 2 doses, which will be approximately 6 weeks to 8 weeks apart.
Got it. And long term, how do you see this OCU200 being positioned as a standalone therapy, and what stage of disease progression patients?
So in the phase 2 portion of the study, if the safety profile looks good and we get the efficacy signal, then primarily we are going to assess this molecule in the non-responders and partial responder. And if the efficacy looks promising in wider group of patients, like with those who are responder as well as non-responder to current therapy, then we'll develop OCU200 as a standalone therapy. However, if we see that there are a specific group of people who respond better to OCU200 compared to the other approved therapy, then we may consider about combination approach in future if required. So it will all be driven by the outcome of the our early, phase 2A or 2 efficacy signal in the wider group of patients, non-responder and responders. Yeah.
Mm-hmm. And given different mechanisms of action, it seems logical, right, that the combination potentially could provide additional benefit versus either one of the therapy alone. Would you generally agree with that? And again, of course, you'll have to see how the data look.
I will have to see, but partially I agree. And the reason I will take this question. So it is not that the integrin pathway doesn't modulate VEGF pathway. Integrin pathway also modulates the VEGF pathway. But the way anti-VEGF therapy works, they are more like a decoy. So if you have a more VEGF, it is a decoy, it will capture. So whatever bad effect you have because of VEGF, that is basically reduced. But the way integrin pathway works, that it basically decreases the level of VEGF, which in the media itself. But so it is slow acting. It is not like a decoy that which captures. So we are hopeful that if this is how it is going to modulate the disease, then OCU200 not only going to work on other pathway linked with the disease, but also it will down-regulate the VEGF secretion. So basically, you will be addressing the cause of disease rather than eliminating the effect of disease by eliminating the VEGF. Okay, but yeah, of course, that learning will come from clinical development, and at that time we'll make decision. Yeah.
Got it. That makes sense. And while the initial focus is on DME, you also mentioned that you have plans to expand into diabetic retinopathy and wet AMD with this molecule. So wanted to ask how you're thinking with respect to timing on initiating these programs. Do you plan on waiting for the DME readout and then taking that into consideration for development of other programs?
That's correct. Once I think we have a efficacy signal in DME patient, then we'll expand into other indications. Got
it. Understand. Okay, I think these are all the questions that I had on your OCU410 and 200 programs. I have a couple more general questions to finish this. First, I wanted to ask about your cash position and cash runway, and what do you expect your cash position to, what milestones do you expect you to carry through?
Yeah, I think as of first quarter, end of first quarter, we disclosed that we had 76.7 million cash on hand. And also during this quarter, last quarter, we raised 14.8 million net proceeds from a deal capital rise. So that puts us into runway into third quarter next year based on that.
Got it. And that will carry us through potentially some additional readouts from the other programs as well.
Yes. Yeah, absolutely. I think as Arun stated, we'll continue our OCU400 program and get into phase 3, and we'll also continue our OCU410, which will start dosing patients. Hopefully next year we'll start getting some data out of those, and similar to like OCU200 too. So there'll be a lot of milestones next year based on how many programs are getting into the clinic this year.
Got it. Understand. And finally, the question that I like to leave with is the more general one. What do you think investors or investor community is missing about Ocugen's story?
I think the big miss, I think the last 2 years, obviously, if you look at it, I think the vaccine, we tried to get Covaxin took most of the focus, in the sense, the investor community is always focusing on it, right? So I think what we had to tell, consistently educate investor community is this is a patient-centric biotech. We have 3 different platform technologies. Gene therapy is focused in ophthalmology, cell therapy is focused on cartilage repairs, and we also got into next-gen vaccines, which are based on inhalation, so which we believe we're the first company to attempt to get into that focus, so that you have vaccines for COVID or flu, which can help to not only give systemic antibodies, but also mucosal antibodies to kind of protect better and also reduce the transmission of the disease. So I think as a company, obviously, these technologies we're developing, in next 3 to 5 years, we have major milestones coming up. The first program, actually, our goal is in next 3 to 5 years, if you take gene therapies, if everything goes according to our plans, we're expected to file the BLA, first BLA in 2025. That means starting 2026, and we'll start launching, gene therapies, followed by, our vaccines and cell therapies. So the vaccines part, as we said, with the limited capital we have, we're efficiently managing it. The focus is on gene therapies and ophthalmology space, followed by cell therapies. And we are seeking government funding for our vaccines at this stage, inhalation vaccines. And they're very novel and they'll have a lot of benefits. So if you do get the government funding, obviously, some of those vaccines will also get launched next 3 to 5 years. So in a nutshell, Ocugen has very strong pipeline with the 3 distinct platform technologies focused on gene therapies, cell therapies, and vaccines. And next 3 to 5 years, we are expected to file, several BLAs and start launching these products and start serving patients globally. That's the plan.
Got it. Excellent. Thank you. I think with that, we are going to finish our session. Again, thank you, Shankar. Thank you, Arun, for joining us. It was great having you at our virtual summit. Thank you. Appreciate your time.
Thank you for having us today.